Assalamualaikum and a very good day to panels. My name is Nurul Ayuni binti Ahmad. My metric number is S50763. Today I would like to present my current research entitled Xestospongia associated microbial community in Bidong, Perhentian and Kapas. For introduction, for marine sponge, they are efficient feeders. They have primary and secondary metabolite and bioactive properties inside the sponge. Hence, sponge associated with microorganisms promising as pharmaceutical leads. They produce novel antibacterial as sponge associated with marine bacteria. The sponge have a well-developed innate immune system. They are key element of biogeochemical cycles in coral reef. For Xestospongia species, they are barrel in shape in the center with a large cavity. The sponge characterized by the presence of finger-like structures on the outer surface. They have a high microbial abundance sponge found on Caribbean coral reef. The color of Xestospongia species maroon to pink. Out of the water, it appears brown or light pink. They have lamella, massive, surface strongly hispid with abundant brushes of larger spicules. The relationship between marine sponge and some of their bacterial and archaea symbionts are bacterial symbionts may play roles in digestion, waste removal, and chemical defense for the sponge host, the involving of antimicrobial activities. For the archaea members of sponge symbiotic microbial communities, they may play an important role in the nitrogen cycle of the ocean. However, there is a scarce on paper reports journals on Xestospongia species-related bacterial and archaea community in Malaysia. There is a little information exists about the taxonomy affiliation of sponge symbiotic microorganisms, especially in archaea. So the objective of my study is to compare bacteria and archaea community with Xestospongia species among 3 islands, Bidong, Perhentian, and Kapas Island by using the natural gradiential electrophoresis, or we know as DGGE. For the methodology, we go for 3 sites as Bidong Island, 15 September 2020, Perhentian Island on 24 September 2020, and Kapas Island on 30 September 2020. After we go for sampling, we went to our lab for sample processing. We will wash the sponge 3 times with sterile artificial seawater to remove unneeded organism, attach the sponge. Then we will cut the sponge and preserve with DNA buffer. Then we will run for DNA extraction by using CTAP method. We run for PCR for DGGE by using 30 GC clamp with universal primer. When the band present, we will run for DGGE by using 6% polyacrylamide gel with 2 type of concentration, low and high concentration. When the band present, we will run for analyze alpha and beta diversity. So the result are for water parameter, DGGE, and alpha and beta diversity. My first result on bacteria for sponge and water sample. The primary use is 338 FGC and 907R, which are in vitro before region of 16S. As for microbial community, by using alpha and beta diversity, we will have Simpson, Shannon, and Magalhae. For Simpson, we used to measure of diversity which into number species that present. Then for Shannon is to characterize the species diversity in the community. For the Magalhae, we want to characterize and we want to calculate the species richness within the area. And this is my second result on bacteria for sponge and water sample by using 338 FGC 519R primer in vitro region of 16S. For discussion, sponge sample, Xestospongia species, and water sample, both bottom and surface were tested positively to contribute of bacteria for present in Bidong, Perhentian, and Kapas Island. The natural gradiential electrophoresis or DGGE we use as molecular technique for rapid analysis of microbial community composition, diversity, and dynamics. For the DGE result, for first running DGE, I used 238FGC907R, which are in V3 before 16S region. The view are less band drivers. V3 before 16S region will maximize nucleotide heterogeneity and display the maximum discriminatory power. For second running DGE, the primary use are 338FGC519R, which are in V3 16S region. Many view of band drivers. V3 16S region gives the highest species richness and distinguish of bacterial species to the genus level. For the DGE result revealed that a large number of band patterns that were attributed to different bacterial species of each bacterial community in both surface and bottom sample throughout 3 different locations. Meanwhile, less band pattern distributed for sponge Xestospongia species. Band pattern for water sample Kapas Island was different because the location is near mainland compared to Bidong and Perhentian Island. So as conclusion, the band pattern of the sponge Xestospongia species samples were less diverse than the seawater samples. As Xestospongia species samples among 3 islands show similar band pattern, whereas seawater samples for both bottom and top at Bidong shows different pattern with those from Perhentian and Kapas Island. V3 16S region is the best to use as give highest species richness and distinguish all bacterial species to the genus level. That's all from me. Thank you.